Hi, I'm Rebecca, and with me are Michael, Sadisha, Tom, and Gwyn, Searles Acme Corp. We're here to discuss how a closed system phosphorus approach can lift bean performance and cut nutrient losses, supporting sustainable farming, minimising phosphorus mining, and safeguarding Moreton Bay and its inhabitants. Phosphorus is a critical component in sustaining global food production, but today's linear P economy, mine, use, waste, creates inefficiency, pollution, and supply risk. A circular phosphorus economy that recovers, safely recycles, and uses phosphorus more efficiently is argued to advance food security and environmental goals. Recycle-based fertilizers come from manures and composts, struvite from municipal wastewater and runoff water, and sewage sludge ash. These sources can supply plant-available phosphorus, co-deliver other nutrients and organics, and partially to fully substitute for mineral phosphate rock, which is a finite non-renewable resource for which reserves are geopolitically concentrated in Morocco and the Western Sahara. Thus, by removing this dependence through recycling, not only do we advance food security and environmental sustainability, but also reduce dependence on dwindling import. Reducing pea losses to water matters locally. See here pictured as a healthy Halophilus spinulosa seagrass meadow from the eastern banks of Moreton Bay. However, Moreton Bay's seagrass meadows, particularly in the Western Bay close to mainland Australia, remains less healthy and highly sensitive to nutrient and sediment loads associated with rainfall and floods, potentially causing toxic algal blooms. Though management has improved Western Bay water quality, vulnerability persists, especially given the IPCC projection of increased heavy rainfall and river flooding by 2050. When excess phosphorus runs off farms into Moreton Bay, it can trigger toxic Lingvia algal blooms and drive eutrophication. When seagrass dies off, it affects the health of the entire ecosystem. Seagrass functions as a nursery habitat for juvenile fish, prawns, and crabs, supports numerous fishery species, and is a major food source for endangered dugongs and green turtles. This is why keeping pea on farms is not only critical for sustainable farming, but also critical for the health of ecosystems downstream. In our research project, we sealed bags around planters to prevent leachate, deliberately modelling a keeping pea in the system approach. On farms, analogous practices include precision crop placement, fertiliser rate control, and riparian buffers and wetlands, which have been shown to cut pea exports more cost effectively than downstream remediation. Therefore, our study tests two CPE-relevant ideas in Phaseolus vulgaris, the common bean. Hypothesis 1, whether a recycled nutrient input, Searles organic pellets, improves growth and chlorophyll status versus a no-pellet control, and Hypothesis 2, whether pairing pellets with phosphorus-free micronutrient solution adds further benefit. These hypotheses align with the literature on recycled fertilizer efficacy and loss prevention, and our bagged pot design reflects the practical goal of keeping pea on land where it delivers value rather than fueling eutrophication.
For the method, we started with 8 bean seedlings in individual punnets, split into 4 groups of 2 using a 2x2 factorial design, testing water type and fertilizer versus no fertilizer. The setups shown on screen below. Punnets were placed in collection bags to retain drainage, creating a closed system with minimal nutrient loss. 1 gram of Searles pellets were added once at the start, 1 centimeter below the surface, around the punnet edge, keeping space from roots to avoid burns. Plants were maintained at laboratory conditions, and each Tuesday and Friday were watered with their assigned solution until drainage appeared. For the reoccurring steps, measurements were taken weekly on Tuesdays, recording height in centimeters, leaf number, first internode length in millimeters, and SPAD, which is leaf greenness. All can be seen at the bottom. For the final measurements, we repeated the same measurements as before, but we first prepared 8 labeled paper towels, removed each plant carefully, washed and dried roots, then cut plants into roots, shoots, leaves, and flowers or fruit. We recorded the wet biomass for each part. We then conducted a two-way ANOVA and measured significance, interactions, and assessed numerical results of the data between the control and treatment plants, which Tom will discuss details now.
Figure 1 and Figure 2 depict how average SPAD and leaf count changed over time. For Figure 1, a lower SPAD reading is depicted for the control groups. For Figure 2, the average leaf count was highest for the control receiving the micronutrient. Compared to the control with tap water, it is significantly higher. Both pellet groups did not differ due to micronutrient water. This suggests that an interaction term is present. Figure 4 shows how plant height differed over time. This, along with the other temporal graphs, show how the individual plants changed over time, and how they were also phenotypically similar at the start of the experiment. This is crucial to reduce variance. Figure 3 and 5 depict the weight on relevant ratios of plant biomass. Root biomass was the highest weight. However, plants treated with fertilizer and micronutrient solution also feature flowers and fruits. This suggests that fertilizer and nutrient solution both have the required nutrients for fruiting and flowering to begin in plants. Figure 6 showcases how the water type influenced SPAD readings in plants, with higher greenness in micronutrient solution treated plants. Figure 8 shows how the pellet type affected SPAD reading, with fertilized plants exhibiting higher readings. This influence on leaf greenness was found to have an independent effect from the water type. Figure 7 shows how water type influenced the leaf count, with higher leaf counts found on plants with micronutrient treatment. Figure 9 shows how stem weight increased if plants were initially fertilized. Figure 10 shows little difference in stem weight for plants fertilized with pellets. Non-fertilized plants' stem weight increased significantly more when treated with micronutrients. This suggests that the initial pellet treatment nullifies the need for further micronutrient application. Figure 11 shows how leaf count was influenced by the interaction between water and pellet type. Fertilised plants face no significant change in leaf count, even when treated with micronutrient solution compared with tap water. The non-fertilised plants exhibited significantly higher leaf counts when treated with the solution. This supports the interaction between water and pellet type. The number of replicates per group was small, 8 plants total split between 4 treatment groups. A significance level of p less than 0.1 was used as preliminary evidence of correlation. While this does weaken the power of the results, it instead allows initial trends to be identified.
Our findings show that organic fertilizer has an increased effect on plant biomass and plant greenness, two indicators that suggest increased plant health. The literature agrees with our findings, with a small increase in phosphorus contributing to greater leaf greenness in Searles et al., an increased phosphorus showing an increase in stem biomass in Pedro GC et al. This fulfills our first hypothesis that organic fertilizer with supplemental nutrients can assist in a system where we recycle our phosphorus. While Searles fertilizer is not a whole alternative to all artificial fertilizer, it is a step in the right direction to stop relying on chemical phosphorus fertilizer to grow our food.
So, to wrap up, what did we learn from this experiment? We found that micronutrient negative pea water makes leaves greener, with significantly higher SPAD values than tap water, and that Searles recycled pellets increase stem mass. We also saw interactions between water type and pellet use, showing that they shape each other's effects. These outcomes align with existing knowledge that greater nutrient and phosphorus availability enhances plant growth, reinforcing the value of this limited resource and the importance of a circular phosphorus economy. Our study clearly identified effects on SPAD and stem weight, though some results were marginal due to short time trials and a small sample size. A longer-term follow-up with more replicates could strengthen these findings. Beyond our pots, this matters because phosphorus runoff drives algal blooms that harmed seagrass in places like Moreton Bay. Our no-leak pot model showed that keeping nutrients in can still boost growth, principles that can scale up on farms through recycled fertilizers and a better nutrient management. So, the big takeaway is simple. Recycled pellets build biomass, micronutrients power the canopy, and together they keep phosphorus where it does the most good, for crops and for ecosystems like Moreton Bay.